So today we're going to talk specifically about blood typing. So you may have learned a little bit about this in some of your science classes or your biology classes, but today we're going to talk about blood, the contents of it, and specifically red blood cells. Next slide, please. So what is blood? So when we talk about blood, we're talking about what comes out of your arm and goes in the tube. And it's made up of a lot of things that this graph can show you, but it's made up of major components. 1%, less than 1% is white blood cells. About 45% of that tube is red blood cells. Less than 1% platelets, and about 55% plasma. So white blood cells, does anybody know what they do? If so, put it in the chat. Red blood cells we're going to talk about today carry oxygen. Platelets do clotting. So whenever you cut yourself, you get a paper cut and you don't bleed out, is because platelets go to that area to help close that wound off to protect yourself. And plasma is pretty much everything else in your blood. So it's liquid, it's water, it's electrolytes, it's proteins, it's minerals, and vitamins. So plasma is everything else that makes up the liquid that's in the tube. And then, good job, Raya. So white blood cells is about 1%, and they are responsible for fighting viruses or bacteria or any kind of illness in our body. So on the bottom of this graph, you'll see basophil, neutrophil, eosinophil, monocytes, plasma cells. All of those things make up your white blood cells, which are responsible for fighting off infections in our body. Next slide. So today we're going to talk about red blood cells. They're also called erythrocytes, if you've seen that word before. So red blood cells look like the pictures that you see, and they are anucleate, which means without a nucleus. Biconcave, which means bi is the prefix is to, and concave means it goes in. So if you were to look at it from the side, it kind of looks like a donut without the hole pushed all the way out of it. So it's a jelly donut that's just got all the goo squished out. And I've had a question, when you give blood, how do they separate the plasma? So there's machines that will basically take the tube of blood that comes out of your arm and they spin it really, really fast. And it takes out the parts of your blood that's not required for plasma donation. So they have really fancy machines that get out the part that they need to donate plasma, and then everything else that is in your blood that's not required for plasma donation is removed from the sample. So erythrocytes and red blood cells are wholly responsible for carrying oxygen to tissues from our lungs and then transporting carbon dioxide, or CO2, from our tissues to our lungs, which we then breathe out. So you breathe in oxygen, that goes into your lungs. Red blood cells pick it up from your lungs. It travels to all the tissues in your body, so from your brain all the way down to your pinky toes, to deliver that oxygen, because our tissues need oxygen to survive, and then picks up carbon dioxide from those tissues, takes it back to the lungs, and that's what you breathe out. So on this slide, you can see the hemoglobin molecule. So hemoglobin is a protein, which is that purple and blue mixture, and that has a ion or a chemical on it called heme, which is the little chemical structure that you see. And heme is responsible for carrying oxygen. So there are 4 hemes per hemoglobin, and each heme can carry 1 oxygen. So how many molecules of oxygen can be carried on a hemoglobin? What do you think? 1 per heme, 4 hemes per hemoglobin. You can carry 4 oxygen molecules per hemoglobin, because each heme can carry 1 oxygen, and there's 4 hemes per hemoglobin. So you can carry 4 oxygen molecules per hemoglobin. And so the reason that red blood cells look the way that they do is because when red blood cells are immature, so whenever they are in the process of maturing to become a functional red blood cell, it has a nucleus. So it looks like a jelly donut that has the jelly in it. And as that red blood cell matures, it expels the nucleus. So it squirts all the jelly out. And that's the reason that it looks why it's biconcave. So mature red blood cells do not have a nucleus. So you may hear in a lot of your science classes that every cell has a nucleus. Well, you can always tell your teachers now that your red blood cells are actually an exception to that, and they don't have a nucleus. Next slide, please. So today we're going to talk about typing specifically. So all red blood cells have types. So we have different blood types, and those are determined by what's called a glycoprotein. So a glycoprotein, all that means is a protein plus a carbohydrate. So you'll see the little globs on the outside of the cell. So where the red arrows are pointing, it's a protein and a carbohydrate or a sugar, and that determines the type. Next slide. So the blood types that we're going to talk about today are A, B, AB, and O. And in the last slide where we talked about the glycoproteins, I'm now going to refer to those as antigens. So that's the same thing, it's just called antigens when you're talking about blood typing. So the antigen on a red blood cell surface determines what type it is. So in type A blood, so the first column, you're going to see there's antigen A, it's on its surface, so the blue circles. And then in type B blood, the purple triangles is antigen B, so it's type B blood. For AB, you have both, so that's the third column. And then for type O blood, you have neither. So antibodies, does anyone know what antibodies do? What are they responsible for? So if you look at the antibodies in the plasma, you'll see that they have what I call like little hands. So they're little hands, you either have triangle hands or you have circle hands. And those triangles or circles will match an antigen. So you'll see that if it has circle hands, it would match a circle antigen. Or if it has triangle hands, then it matches a triangle antigen. So if you look at the names of the antibodies, you have anti-B antibodies with the triangle hands, which means that matches antigen B. So what happens with antibodies is if it finds its antigen. So if I am an anti-B antibody with my triangle hand and I find my B antigen, I'm going to find it, I'm going to clump up, and I'm going to take it to the immune system to be destroyed. So antibodies are responsible for finding specific antigens, that's why they have this unique little hands, to then take it to the immune system to be destroyed. They're like basically on patrol for very specific things to make sure that you don't get sick. So in blood typing, you have antibodies in the plasma of your blood. If your antibodies are responsible for finding its specific antigen to then take to the immune system to be destroyed, it wouldn't make much sense for your antigens on your red blood cell surface and the antibodies to be the same, because it would find its antigen, take it to the immune system, and be destroyed. And we don't want our red blood cells to be destroyed because they are responsible for carrying oxygen to tissues. So in blood typing, you will see that whatever antigen is present on the red blood cell surface, so the first row, you will have the opposite antibody. So that's the second row, the plasma. So if you have the A antigen, you're not going to have the A antibody because they would find each other and go to the immune system and be destroyed. So in type A blood, you have the A antigen present on the red blood cell surface. You have anti-B antibodies, which is the opposite. And then in type B blood, you have B antigens on its surface, and then anti-A antibodies, the opposite. Now in type AB blood, you have neither antibody because you have both antigens. So it's the opposite. If you had either anti-B or anti-A antibodies, it would find its antigen, take that red blood cell to the immune system and destroy it, which again is really bad because our Red blood cells are responsible for carrying oxygen. We don't get oxygen to our tissues, those tissues die, it's bad news bears. And then in type O blood, we have neither antigen on its surface, on the red blood cell surface. So that's the very last column. So you're going to have both antibodies. What questions do we have about this chart at all?
Is that why it's bad to get injected with the wrong type of blood because the antibodies would destroy it?
Yes, so we're going to talk about that in a little bit, about blood donation and universal blood types and universal blood or donors and recipients. So yes, you have to make sure that the blood that you receive matches, so is compatible with what you have, because otherwise your antibodies would attack that donor blood and destroy it, which if you need blood, you don't want that donated blood to you to be destroyed, because that would defeat the purpose. You would not get any better. Any other questions about this? All right, next slide, please. Okay, so blood types are inherited. And so the glycoproteins, like we talked about earlier, which is the antigens, they determine your blood type, and it's inherited from your parents. So you get one allele, which is a form of a gene, from your mom, and you get one allele or a form of a gene from your dad. And those alleles come together to make the blood type that you will have. So does anyone know the difference between genotype and phenotype? If so, go ahead and put it in the chat. So the difference, so you have the 4 different blood types. We have A, B, and O. And then there are different genotypes or combination of alleles that can make a certain phenotype. So A and B are dominant. So that means if you have at least one copy, either from mom or dad, that's what shows up. And O is recessive. So O, you have to have a copy from mom and a copy from dad. And then there's also what's called codominance in blood typing. So codominance, which you might think of as like co-presidents, you both show up. So you're both in that role. And that is what type AB blood is. You get an A, you get a B, they're both dominant and they both show up. So genotype is the combination of alleles that is in your genes. So that's what you get from mom and what you get from dad. Whatever combination that is your genotype. And your phenotype is how your genes are expressed. So it's a physical representation of it. So your genotype is the combination of alleles, and your phenotype is how it's physically represented. So again, so for example, type A blood, you can get 2 copies of A. So you get one from mom and one from dad. Or you get an A from one parent and an O from one parent. So what that means is because A is dominant and O is recessive, if you get an A, you're going to show up with at least type A blood. Same thing with type B. You can either get a B from both parents or a B from one parent and an O from the other. And because O is recessive and B would be dominant, you have type B blood. That's your phenotype. And again, type AB is because A and B are codominant. So you get an A from one parent and a B from one parent, you get AB blood. And type O is recessive, so you have to get an O from each parent. What questions do we have about this? All right, if you have any questions, you can feel free to put them in the chat. Next slide, please. So this is just a little experiment. So this is, if you've ever seen a Punnett Square, this is my PowerPoint version of a Punnett Square. So we have an allele from mom, we have an allele from dad. You get, your mom is, her genotype is type AA, so her phenotype would be A. She has type A blood. Dad's genotype is BO, so his phenotype is type B. So he has type B blood, mom has type A blood. So these two parents are going to make offspring, and Dad can give a B or he can give an O to his offspring. Mom can give an A or she can give an A. That's all she's got. Can these parents have a kid who is type B? So in this graph in the middle, you're going to see the possible combinations of alleles based on these two parents. Can they have a kid who is type B? Yes or no? So correct. No, they cannot. Can they have a kid who's type O? Again, correct. No is the answer. So what they can have a kid who's type AB, which you can see in the squares in the middle. I'm pointing at my screen, but obviously you can't see that. Or they have a kid who's type A. So which parent is the problem? If they could have a kid who is type O, which parent's genotype would have to change, mom or dad? So remember, to have a kid type O, each parent has to give an O. So we've got one parent who's got an O, that's dad. Mom doesn't have an O to give. If mom had an O to give, they could have a type O kid. Great job. Y'all doing great. Next slide, please. So there was a question, is there a reason that O is recessive? I don't know. There might be a reason, but I'm not sure why. I just know that it is, and that it's more rare to have type O blood because you have to have a very specific combination of alleles to get type O blood. But I don't know why specifically it is recessive. It's a good question. Okay, so Rh factor, that's from the rhesus monkey. That's what determines either to have a positive blood type or a negative blood type. And there's antibodies and antigens exactly the same as what's in ABO blood typing, or what we just talked about. What we just talked about is technically called ABO blood typing. So for some reason, for Rh factor, it's called the D antigen or the anti-D antibody. So it's not the R antigen. I have no idea why. That seems to make a lot more sense to me, but it's called the D antigen. So just like on the ABO blood typing that we already talked about, with positive and negative blood typing, if your blood type is positive, you have the D antigen, so it's present on the red blood cell surface. If it's negative, you do not have the D antigen. So if it's positive, you have the D antigen. If it's negative, you don't. But if you have Rh negative blood, so you don't have the D antigen, again, the rule of opposites for antibodies, you're going to have the anti-D antibody in Rh negative blood. So it's the same principle. If you have the D antigen, you don't want to have that antibody because it's going to find its antigen and destroy the red blood cell. So if you have the antigen, you don't want the antibody. But if you don't have the antigen, you do want the antibody because it's the opposite. Any questions about this? Okay, next slide, please. So Rh positive, so positive blood is dominant. So if you get at least one copy from either parent, you will have positive blood. The only way to have negative blood is to have a negative from both parents. So it's possible if you look at the bottom table, that you can have two parents that have positive blood types to have a negative blood type child. And the reason being is that both of those parents would have to have a positive and a negative. And so then both parents would have a negative to give, and so that child could have a negative blood type. But Rh positive is dominant. It's pretty much all you need to know. Next slide. So why do we care? Why do we care about positive and negative blood types? So there is a disease called Hemolytic Disease of the newborn, or erythroblastosis fetalis. So if you look at the fancy name at the bottom, erythroblastosis, erythroblast means immature red blood cell, and fetalis is fetal, so that means babies. And hemolytic, if you've ever heard of something that has the LYTIC at the end, It means that something is destroyed. So what happens is that an Rh negative woman and an Rh positive man conceive a child. That means that that child is going to be positive, because positive is dominant. Positive and negative, that means positive is going to show up. So mom is now pregnant with an Rh positive fetus. During pregnancy, lots of times mom and baby's blood mixes. When mom and baby's blood mixes, mom becomes what's called sensitized. So these green diamonds in this picture form, because just like if you get the flu or you get a cold, your body arms itself so that the next time you encounter that flu or that cold, you don't get as sick. You're going to make antibodies to make sure that you can fight off that infection if you ever get it again. But instead, that process happens when this woman is pregnant, because that baby's blood is not compatible with hers. When their blood mixes, either during childbirth or during the pregnancy, mom forms the antibodies to say, oh no, this blood is foreign to me. This blood is bad. It doesn't match mine. Next time we see it, we're going to fight it. So she becomes pregnant again with another Rh positive fetus. And those antibodies then attack the baby's red blood cells. And that's what's called hemolytic. So the antibodies attack the baby's red blood cells because it's going to fight those red blood cells because it doesn't match mom. And the baby is then born, if it's born, is born with immature red blood cells. So that's the erythroblastosis. Those immature red blood cells cannot carry oxygen. So our red blood cells are responsible for carrying oxygen to our tissues. Our tissues have to have oxygen to be able to survive. So the baby's blood cells cannot carry oxygen. So obviously this is very bad for the baby. But we have a way to basically prevent the woman becoming sensitized or the antibodies forming. We have a way to prevent that, because people don't know each other's blood type. You don't know that your partner is going to have a compatible blood type with yours. So there are shots for women who are Rh negative that are called RhoGAM, which you might have heard before. So you get that, I think, 2 to 3 times during your pregnancy, and it prevents the antibodies from being formed. So it doesn't matter what the father's blood type is. If the woman is Rh negative, she gets those shots when she finds out she's pregnant, and it prevents this process from happening. So you can carry the pregnancy to full term. You have the baby's blood's perfectly fine. Any questions about this?
Could that result in the woman getting sick because she doesn't have antibodies to like fight a virus?
So these antibodies are specific to the Rh positive red blood cells. So it's not going to fight a flu or a cold. These antibodies would specifically form to fight that foreign blood. So it wouldn't affect mom in any other way, because in any other situation besides her being pregnant with a fetus that doesn't have the same blood type as her, she would never encounter that. She would never encounter that blood anyway. So it's perfectly normal for mom to get those shots if she's Rh negative, and doesn't harm mom or baby in any way. Now, if you're an Rh positive woman, so if you have a positive blood type, you don't have to worry about it. Any other questions about this? If the antibodies are already sensitized, is it possible to cure or desensitize them, or does the treatment have to come before? That's a really good question. So I don't know. So the reason that RhoGAM works really well is because, so once a woman finds out she's pregnant, they immediately will blood type her. So once she finds out she's pregnant and they blood type her, they will know if she's positive or negative. If she's positive, they don't have to worry about doing the RhoGAM because the sensitization won't happen, because baby's blood will match mom. But if she's negative, then she immediately will get RhoGAM. Now, if something were to happen before where she had a complete pregnancy, did not get blood tested, and didn't have any kind of medical intervention during that pregnancy, I'm not sure what would happen with a subsequent pregnancy, it seems like this process would happen. I don't know if there's another cure or treatment to prevent this process from happening, or, and I'm like I said, I'm not sure, or if you got RhoGAM the second pregnancy that it would work. That's a really good question though. All right, next slide, please. So the concept of universality, this is about universal donors and universal recipients. So if you look on this graph, you're going to see that if you can donate to somebody whose blood type matches exactly to yours, right? O negative can donate to O negative, A positive to A positive, so on and so forth. But there are concepts of universality for recipients and donors. So if you know the universal blood type, if the universal donor blood type, type it in the chat. So universal recipient blood type is AB positive. So you'll see that on the grid at the very bottom row where all the checks are all the way across. That means AB positive can receive any blood type. And then, good job everybody in the chat, O negative, which is the columns, can donate to anybody. So O negative can donate to any blood type, including itself, obviously, O negative. And AB positive can receive blood from any blood type. So the reason being is that if you think of certain scenarios where, for example, my blood type, I'm a recipient, my blood type is AB positive. I cut my arm off and I'm bleeding out and I need blood because my tissues need oxygen, and that's what blood does. And I have AB positive blood inside my body, but I need more blood to be able to do what I need to do to survive. My recipient type, AB positive, if this is a red blood cell, I have the A antigen, I have the B antigen, and I have the D antigen, AB positive. Does that make sense? So if you think about what antibodies do I have in my recipient blood type, I have every antigen. What antibodies do I have? Every antigen, so it's the rule of opposites. If I have all the antigens, I have no antibodies. And the reason that makes me the universal recipient is that regardless of what blood type is donated to me, my antibodies, I have no antibodies to fight off that donated blood. So whatever type is donated to me, I have nothing with which to fight. Because that's what antibodies do. Antibodies find something that they recognize that shouldn't be there. They take it to the immune system to be destroyed. But since I have the A antigen, B antigen, and D antigen, I have 0 antibodies in my blood to fight that donated blood to me, which is good, right? I don't want to fight my donated blood because I need it to survive. Now, the opposite is true for the universal donor. So let's say that I chop my arm off and I need blood again. And my blood type is B positive. I can receive O negative blood because that blood that's donated to me, this is a red blood cell, does it have an A antigen? No, because it's type O. Does it have a B antigen? No, because it's type O. Does it have a D antigen? No, because it's negative. So this completely naked blood cell can be donated to anybody because regardless of what the recipient's blood type is, regardless of what their antibodies are, there is nothing on this red blood cell for an antibody to attack. There are no antigens on its surface. So it doesn't matter what antibodies I have, there's nothing on this red blood cell that I can attack. So O negative is the universal donor for that reason, because there's nothing on its surface that an antibody can attack. And AB positive is the universal recipient because there is nothing in my recipient blood to attack with. All right. What questions do you have about the concepts of universality? Okay, if you have any questions, put them in the chat. We can go to the next slide. So if we were going to do this lab in person, you would get a card that has four circles on its surface. It has what's called the blood sample or the control, anti-A, anti-B, and anti-D circles. So a control in a science experiment is just a comparison. Nothing's going to happen. So you would poke your finger, you put some blood on the circle, and nothing's going to happen. It's just a comparison. On the anti-A, anti-B, and anti-D circles are dried antibodies. So what happens, what you're looking for when you do a blood typing kit is you look for an agglutination reaction. So what does agglutination sound like? What word is in agglutination? Glucose, kind of close, yes, but glue. Things stick together. We're just looking for clumps. So we're looking for a reaction between antibodies and their specific antigen. We're looking basically for this. So when it clumps, that means an antibody has found its specific antigen. So that's why you'll see some circles have clumps and some circles don't. Because if there's clumping, there is a reaction where an antibody has found its antigen. So if you see clumping in the anti-A circle, that means the A antigen is present, because that means that antibody found its antigen, they clump together. If there's clumping in anti-B, that means the B antigen is present, because that antibody found its antigen and they clump together. If there is clumping in anti-D, that means it's positive, because the anti-D antibody found its D antigen, which is only present on positive blood. So we're going to go through the first row. We're going to read these blood types. So the first row I will help you with. So the blood sample is just a comparison. There's clumping in anti-A, so it's got the A antigen. There's no clumping in anti-B, so there's no B antigen. And there's clumping in anti-D, which means it's positive. So the first blood type is A positive. So what about the second row? Put in the chat, what do you think it is? Oh yes, a bunch of genuses. Yes, B positive, because there's clumping in B and there's clumping in D, so it's B positive. What about the third row? This one's a little bit trickier. Yes, yes, yes. A, B positive. We have clumping in A, we have clumping in B, we have clumping in D. So it's A, B positive. Has all of the antigens. And then the last row, what is that? It's the exact opposite, correct. So there's no A, there's no B, so it's got to be O, and there's no clumping in the anti-D column, so it's negative. Next slide. Look, you're all geniuses. Good job. Okay, so that's the end of this presentation. Does anybody have any questions about anything in the whole wide world?
Guys, if you don't have a question about blood typing, if you want to go into a medical career, this is a good time to ask Misty questions about career opportunities, or maybe what education is required to do a certain job or something like that. If you have any questions, she can help you answer that.
Yeah, you can ask me literally anything in the whole wide world.
Do you guys know if hospitals are doing volunteer programs this summer?
That's a really good question. Unfortunately, I don't know anything specifically. I know that with COVID, the restrictions are a lot more stringent with allowing anybody to go into hospitals. So I would check with your local hospital to see if they're doing any kind of volunteer program. I would assume that it's probably pretty limited if they're letting anybody in, just because of the risk of either you getting sick or you bringing any kind of sickness in for the patients.
I'm pretty certain the mission halted all volunteers at a high school level anyway.
Would you need a certain degree to work in peds? Well, that would depend on what you want to do with pediatrics. So that would, if you wanted to be a nurse, if you wanted to be a doctor, if you wanted to be a PA or a nurse practitioner. It depends on whatever kind of career you want to go into, because there are all different types of health professions that work with pediatrics. You know, anything from, like I said, a nurse or a doctor to a physical therapist or a pharmacist or a respiratory therapist. All those different careers will work with pediatrics or kids. So as far as a specific degree to work with them in the healthcare field, no, not necessarily. It would be more training after you decided what career field specifically you wanted to do within pediatrics. Do you know anything about the ribbon program at Western? I know some about it, and if Brian or Amy knows a little bit more, excuse me, they can chime in. So the ribbon program, certain community colleges have a partnership with Western, where you go and you get part of your degree. You start at a community college, and then you go an expedited route right to Western to get your BSN, which is a Bachelor's of Science in Nursing. And so you would finish your career, your education to get your Bachelor's of Science at Western. I'm not sure, outside of AB Tech, I know is one of the schools that has that partnership with Western. If you have any questions specifically about that, I would definitely ask your guidance counselor or AB Tech if that is the community college that you're interested in. And they can definitely point you in the right direction of anything that you would need to do now.
Any other questions?
How does blood typing play into surgery in general? That's a really good question. So if you think about what happens in surgery, so you're cutting somebody open to do something on the inside. Whenever you cut somebody open, just like even if you get a paper cut, you got a lot of blood that comes out. So whenever you are doing any kind of surgery on somebody, you want to know what their blood type is. Because if something were to go wrong or that person bleeds out, so if that person is sitting on your surgery table or in the operating room and they're bleeding out and they need blood put back into them, you need to know what their blood type is. You need to know what types of blood they can receive just in case something goes wrong. So it's very important in surgery. It's very important in OBGYN or anything where you're cutting somebody open. And it's also good to know in other healthcare careers too, because it's a very pretty basic thing, but it really can impact people's health a lot. How long can blood be stored? That's a really good question. I don't know how long blood can be stored. I know it does not have an infinite shelf life. So that's why the Red Cross and other blood donation places are always asking for more donations because it cannot be infinitely stored. And I know that in people, the average life cycle of a blood cell, so a red blood cell, stay around with you from the day you're born to the day you die. It's about 120 days. So every 120 days, you get a whole new set of red blood cells. So they're constantly being regenerated and destroyed. And let's see what others. How do surgeons control how much blood the patient's using? So one is by avoiding cutting major arteries as much as you can. So if you're doing, a surgery on somebody's hand, you don't want to cut their neck and cut their carotid artery where they start bleeding out. And you want to avoid any major arteries in their hand as much as you can to avoid that person just bleeding out. Another way is to do minimally invasive procedures. So for example, if you got your gallbladder taken out, maybe 15 years ago, it would be a really big scar and a really big incision that would have to be made. But now they do laparoscopic surgery, where basically they just put a couple pinholes into your abdomen, stick a robot in there, get your gallbladder out, and you just have a couple stitches and you're out the same day. So other ways to do it is just making sure that using just appropriate techniques or cauterizing or like burning the wound to help stop the bleeding. In a rush, like in an emergency, can anyone almost get O negative blood without checking their blood type for it? So yes, O negative blood is the universal donor. So regardless of my blood type, I can get O negative blood no matter what. So that's why, for example, if you have O negative blood and you go to donate your blood, the Red Cross is going to absolutely love you, because that blood can be given to absolutely anybody. Whereas if your blood type is B positive, you can't give blood to everybody. You can only give blood to a certain amount of people, depending on what their blood type is.
Just to add to that, I'll say I have a friend who works in the emergency department in Johnson City, and he said that they have 2 trauma bays, and in between the 2 trauma bays, they have a cooler that has O negative blood in it. So if they have someone that comes in with a lot of trauma and they've lost a ton of blood, they'll hang up those bags really quick, and they have an infusion or a thing that infuses and pushes that blood. I think they can push an entire bag in a minute and a half. So it's pretty incredible, but they don't take the time to check your blood type. They'll just give you O negative until they know your exact blood type later on. And 42 days is the shelf life of blood. I Googled it.
Okay, guys, any more questions?
You missed one here, Misty. How do surgeons control how much blood the patient is losing? Did you mention that one?
Yes, you
I
did.
think I did.
Oh.
Yeah, I did.
Okay, maybe I wasn't paying attention.
But
You were
thanks for
Googling.
paying attention.
Okay, so we want to be mindful of everyone's time. I'm going to share my screen, which is a certificate of completion. So if you need to produce that for a teacher, you can just screenshot it or take a photo of the screen. We thank you guys for coming. Next week, we're going to have a pig dissection workshop. So if you're interested in that, join us next Tuesday afternoon.